and I'm doing it because during quarantine I haven't been super motivated to like get up, get ready. It's been kind of a struggle waking up in the morning because I'm like, I'm not gonna see anyone. But I thought this would be a good way to motivate me to be a functional human. Um, so that's what I'm doing. I'm currently working on making a bunch of revisions for my first draft for my paper. I am going into my third year of a PhD program where I'm doing mathematical modeling in biology, and my project's focusing on neuroscience. So I am going to be working on that now. Yeah. So the first thing I'm going to be doing today is I'm going to be trying to recreate this figure. As you can see, I've been working on it. It's been taking way too long. I'm recreating this figure for my PI's paper from like 2008. But this is adapted, so he did this analysis for looking at cardiovascular networks and the branching of blood vessels, and I'm looking at applying some of these mathematical principles to look at branching in neurons. Just to give you an idea, I'll talk more about my project later probably because I can't really explain everything now, but hopefully I can soon. This is an example of the branch projection from a neuron. So this is connected to a cell body, and these branch projections connect to the projections of other neurons. And this is how they communicate with each other. And I want to understand what is driving these structures and branching patterns. And so we have some methods of analysis for this. We're trying to see whether we can relate it back to function. So for example, evolutionarily, you would think maybe neurons are the structures optimized to minimize the amount of time it takes for signals to travel between neurons. So I'm using those principles to try to make theoretical predictions for the structure and then compare it to what I see in the data. So this is just for my paper. I'm in the theory section of the paper. I'm explaining what our basic biophysical theory is. I just have to create this. The reason I'm starting with this is because obviously it doesn't really take that much brain energy to work on. It's not hard. It just takes a long time and it needs to be done. It's one of those annoying things, like you just have to do it. It doesn't really take that much intelligence to do it, but you just have to do it. And I'm not really awake yet, so hopefully this will warm me up. Also, while I'm doing my work, I really like playing these kind of lo-fi jazz instrumental music. It's mostly because if I play something that's has lyrics and is upbeat, I just want to start singing along and dancing, which is not really helpful. But this is pleasant. It makes me feel like I'm in a coffee shop, like the pre-corona times. And I know I have like 10 million tabs open, which is stressing me out too. Okay, so I finally finished the figure and put it in, and I'm really happy with how it turned out. Yeah, so I'm gonna send this to my PI soon to make sure it's fine, but I've been playing around with the colors. So I also tried this one, which was like closer to the color of the fluorescent dye in the actual image. So I was trying to see which one I prefer, but I think I prefer this one because the other one's like, this one's like kind of blinding. So I think I'm gonna go with this one. So yeah, moving on to the next thing. So the next thing I have to do is, so I want to use these, so this is a database called neuromorpho.org. It's really cool. It has a lot of different species of these reconstruction neurons. Sorry, I just like completely blanked out. It has a lot of different types of neurons from a whole range of species, from humans and rodents and what you normally think of in experiments, but also whales, um, chimpanzees, all kinds of just random animals. And this is collected from different papers. Um, and anyone can use it. But basically, they give you these terms of service. And so, if you use this data, which most of my actually, I think all of my analysis is from this, you basically have to cite this paper, cite one of the papers that's associated with each individual neuron. So, if you look at the bottom, you can find that link over here. And um, so I am wondering whether, if I am citing this, if I can use this reconstruction preview from the site, because that is something I would be interested in doing, to show a visual next to my data. I'm doing a kind of a comparative analysis of different types of cells, so I would like to show kind of their visual differences next to the data that I've shown. But I'm not sure whether I'm allowed to use this using these other citation guidelines for using the actual data. So I'm gonna try to see if I can contact the website and see what their policies are on that. I'm now going to answer some emails. I have some fellowship things I have to sort out, so I was on a training grant for the past 2 years of my PhD program, and I have to write a summary of like the things I've done, and kind of like, conclude that. I'm just starting my new fellowship, which I'm really excited about. And I got an email basically, so the fellowship covers most of my tuition, but there's a little part that's not covered. But I am getting, like, the school kind of ends up covering it. Like, I think my department pays for half, and like the grad division pays for half or something like that of the remainder. So basically I have to fill out these forms and like send them back. But it's worth it, because then, it's nice to have money. But you just have to do all the stuff. So I'm gonna do that right now. Alright, so I finished all the administrative things with just like fellowship, things like that, sending emails, and also for the org that I'm part of, I volunteered to be part of the tabling and panels for incoming students, even though I've been telling myself like I need to focus on myself and my work and say no to things more. I felt like it would be good to do that, especially because right now I have time, and it's going to be this month, and then it's before classes start, so. And I got an email back from Neuromorpho, and they basically said that I could just use the images and use the same citation guidelines as the data, which is really good. So I'm hoping that I can just format my figures and then do some literature review. So that's what I'm going to do for the rest of the day, sorry. And I think I'm going to then try to email this version to my PI. So I have quite a way to go, but I think I need to take the dog out for a walk because she's definitely getting pretty restless. I have to introduce you to my dog, so that's the dog. She's really excited to go on a walk. This girl is all done with her walk. She's like, who are you talking to? Okay, so I took the dog out and just threw the laundry in the machine because I'm running out of clothes and I haven't done it in a while. And now I'm gonna do a literature search and find. These are mostly papers I've already read when I was originally doing my research, but I want to find specific things that I've kind of used in the conclusion that I've made in my paper already, but I just want to find where the actual, just some good supporting information. I want to be more clear on that, so that's what I'm doing right now. I'm going to be reading some papers, taking some notes on them here, and then adding them to my citations. So that's what I'm doing right now. So first I'm going to read from papers, and then I'm going to read from my textbook a little, which also cites papers. So basically right now I'm just doing a lot of reading for the paper, which doesn't really feel like I'm actually doing anything, but it is fun. So yeah, I'm gonna turn on the fan. It's like 10 p.m. and I didn't quite get everything I wanted to do done. Actually, like, I'm not ready to submit my draft to my PI, but it was pretty good because I ended up spending a lot of time reading papers. I kind of went down a rabbit hole, where like I would go on one paper and they would like cite another paper, and I was like, oh, I need to see what the original results for this, and then I just kind of went down this huge rabbit hole. I found some papers from like the 30s. I didn't end up using those, but yeah, so it was good because I feel like now, I mean, I've already written my discussion section, and I feel like the stuff I found supports what I already have, but I feel kind of more confident. Like, I feel like when I edit it, I can add more citations and I can add more things that support what I have, which is always good. So tomorrow I'm gonna I have a few more papers I want to read that I've already read and I've already cited in my paper, but I want to make sure I'm not missing anything. So I'm gonna do that, and then I'm gonna read some sections in my textbook that I've marked to read later. And then I'm just going to work on writing those sections. And then after that, I'm going to work on the figures. So I think I have a pretty good plan for tomorrow. So yeah, thank you. If you stayed this whole time, thank you for hanging out with me. And this was really fun. And I feel like I was able to be more productive than I probably would have. And usually I would probably like take more breaks or like go work out and stuff, and I've been wanting to do that, but right now it's really crunch time. Like, I really just want to submit this paper as soon as possible, so I am just trying to get this done. But yeah, well, bye. See you next time.